So, office hours today after lecture in the normal slot, 9 to 10 a.m., are in 2195 VLSB. It's right around the corner on this floor. It's a bigger room. And then next week, so you start the ecology series on Friday, and Professor Roderick will take over the 5th floor office hours room in the Monday, Wednesday, Friday slot, 9 to 10. So next week, Monday, Wednesday, Friday, his office hours will be upstairs, 9 to 10. Mine next week will be 12:30 to 2 p.m. on Monday and Wednesday in 2195. So that'll provide additional time for general questions leading up to the exam on Wednesday night, right? I have some review material here based on emails I received, a small number of emails, and I'll just cover questions from those emails and a couple of other general things from previous lectures and a few final slides for this section. Any questions before I get started? Yes, please.
Have the terms and concepts list been posted yet?
They are up. They've been updated after each lecture, more or less. They're up to date through the last lecture. You have a link on B Courses, on the main page on B Courses. There's a link to that list, and then I think I sent an original email with the link too. Best place to find it would be B Courses homepage. And that's a good guide. That terms list is a fundamental guide to the key terms to know from the slides. If there's a species, an organism's name that I went through in the slides that's not on that list, no need to memorize the details about that organism. Any exam question would give you those details as part of the question. But if there's an organism on that list, like Tiktaalik ended up on that list, or gorilla ended up on that list, yeah, please know who gorilla is, who Tiktaalik is. Yeah. The midterms, I'll talk about the coverage of the midterms at the end of the lecture today. But yeah, basically you can plan to get up to date for the midterm through all the lectures. Otherwise, you're postponing the work until the end of the semester anyways. So you have a good week, but I'll be more exact on that at the end of the lecture. Anything else? Yep.
So you said, so conceptually speaking, all the major cell and the specific example.
No, conceptually speaking, you should be understanding the concepts that are presented in the lectures. So I haven't listed every single conceptual relation in those terms lists. Those are just terms lists presenting the fundamental terms that are required for this section. You're going to be tested, I hope, on your general understanding of evolutionary biology. A good question will ask you to try to relate the terms that you know to the concepts that have been presented. I don't think I can be any more specific than that without passing out the questions. But you have a whole big set of practice questions, right? I put up a second set of practice questions. It's quite long. You are in no way obligated to do them. If they bore you, if you don't feel like you benefit from them, skip them. But many people have asked me for more questions, and they're close to the types of exam questions that I write, although they are drawn from textbook. They're based on textbook questions. Anything more? All right. We finished with a discussion of humans arriving in California, having migrated out of Africa as Homo sapiens. I spoke of a first wave out of Africa of humans, dated to about 1.8 million years ago. Someone asked me, if humans left Africa 1.8 million years ago, how do we find evidence of humans in Georgia at Dmanisi at 1.8 million years ago? Wouldn't it have taken some time to get there? Well, when we say 1.8 million years ago for leaving Africa, it's based on that date at that fossil site. So we don't know exactly when Homo left the continent. There is broad agreement that Homo evolved on the African continent and not in Asia, for example. But the exact timing of these events is rough. 1.8 at Dmanisi, sure, it was some time before that people actually left. The fossil record's not that precise. Then, in Africa, Homo having come to occupy Eurasia generally, Homo's continuing to evolve also in Africa. And Homo sapiens, this creature that's anatomically like us, modern Homo, humans, Homo sapiens, can't be distinguished from us in general in terms of skull features, skeletal features. Homo sapiens evolves in Africa sometime around 200,000, 160,000 years ago. And those dates are based on some archaeological finds at 200,000, some fossil finds at 160, somewhere in there. A couple 100,000 years ago, Homo sapiens emerges with all the attendant features of Homo sapiens: deep conceptual thinking, production of artwork, communication abilities that are unique probably, although those don't fossilize well. Now Homo sapiens, having evolved in Africa, then moves out of Africa in that second wave out of Africa, second major wave, and Homo sapiens makes their way to the Americas very late in this whole process, only 13,000 years ago or so. There's some bits of evidence that point to maybe 30,000 years ago in the Americas, but they haven't held up. 13,000 is a good estimate. See how young that is? See how recent that is in our big scale? California has some of the oldest evidence at 12,000 years ago for people arriving. So I'm going to talk about some of the effects that humans have had in that process, just to finish up. And I'll go quickly so that we can focus on review material. This object is a new type of rock described in a geological journal this year called a plastiglomerate. Now, these things were first, the description of these things was based on items found in Hawaii, on Hawaiian beaches. And they involve plastic melted together with rock and coral and shells and sand into this object that's coherent and durable and is making its way into the ocean sediments and forming part of the sedimentary layers in the oceans, at least off Hawaii and probably around the world. Melted plastic bonding to these other quote-unquote natural materials. So this is named as a new type of rock in the rock record that clearly has a human fingerprint because plastics were invented by us in the, 50s or early 60s or something. So this will play in later when I get more toward the present about human effects on global ecosystems. But let's take a step back to the end of the Pleistocene and see a possible role for humans in shaping ecosystems of California, ecosystems of the world, but California specifically. La Brea, where some of you have probably visited the museum related to the La Brea tar pits in Los Angeles. La Brea is this tar seep. It was a tar seep. Now it's covered by LA. That of this nature, this is, you can visit near in Southern California. We went down with a class. That's a bunch of students up there from Berkeley visiting this tar seep where this tar is still flowing across the surface, sticky. You can go and poke it, and it's sticky, and you'll find snails stuck in it. You'll find a dead cat stuck in it. You'll find a bird stuck in it. Stuff gets stuck in it because it's super sticky, and the more you fight it, the more you get stuck. And if you're a dying prey item, then other animals come and try to eat you, and then they get stuck, and you all end up in this ball of tar. Well, that was in the nature of La Brea back at the end of the Pleistocene, 60,000 to 10,000, 4,000 years ago. 38 maybe is the earliest in that time window. La Brea was first worked by Berkeley scientists, and many of those fossils came here. And many are indeed stored in the Campanile. If if you've heard that on your tour of Berkeley. It's true. Campanile is full of La Brea fossils. Fascinating room up on, the 5th floor or something of the Campanile. Click this button if you want to hear an NPR story that was just produced a couple weeks ago about that. But scientifically, what's of importance is this massive collection of materials showing California history during this time window. Hundreds of species of plants and animals preserved, well preserved, whole skeletons, macro-botanical remains, including dire wolves, which everyone seems to know about these days. A stocky, big wolf, big, heavy, stocky wolf. Our wolves today are tall, but more slender. These things were just robust, heavy creatures. And there are gobs of them from these deposits. Also coyotes and other foxes. So rich in these big predators, California was. Less known are giant short-faced bears, Arctodus, very scary bear. Big bears are formidable, right? So heavy, dim-witted sometimes, singularly focused if their babies are around. You don't want to mess with a big brown bear. Well, get Arctodus, which I think I have a diagram. Here's Arctodus. Here's your little grizzly bear. There's a polar bear. Polar bears are enormous, right? Well, Arctodus stood this tall. There's the artist, Carl Buell, with Arctodus breathing down his face. This thing was present in the Americas when humans arrived, like the dire wolves. Tall, long-legged bear that probably was a good runner in open country. This was a cursorial bear, a running bear, with teeth that were more built for slicing meat. So it was a hyper-carnivorous running bear, very scary bear that was encountered by people. It's fascinating to think about. Big lions, bigger than a big male African lion, present in California during this end Pleistocene time. The saber-toothed cats were here, our state fossil, Smilodon, present in California. A cheetah. This is my favorite, this creature, a big cheetah, big heavy cheetah. Well, quote-unquote cheetah. Ancient DNA has been extracted from these guys. Morphologically, it wasn't clear whether they were true cheetahs or related to mountain lions, local cats. Ancient DNA pulled out of them finds in a cladogram that they're actually Miracinonyx, this American cheetah, is actually related to mountain lions most closely. So it's a big, running, fast mountain lion, essentially, that was present across the Americas. California to Pennsylvania, Tennessee, Florida. This thing was around in the open plains, hunting swift prey, no doubt. Ground sloths. Everyone seems to love sloths. Oh, sloths. You know, they have that permanent smile and they sit in the treetops, all peaceful. Well, there were big sloths. This was sloth country. Big bear-sized sloths. A buffalo-sized sloths. Big things. Ground sloths. There are no more ground sloths. All gone. Well, where did they go is an interesting debate. So at a place like La Brea, your herbivore group, your carnivoran group, mostly gone. Bison still survive elsewhere, not here. Horses, horse relatives. Camels, this was camel country. Camels like llamas and guanacos, you know, those types of camels. Camels were present all over North America, all gone. So the debate centers on whether there's a human hand in having eliminated these things in some way or contributed to their disappearance, or alternatively, whether climate change, some natural, quote-unquote natural mechanism is at play. Richard Owen, old Richard Owen, Darwin's contemporary and antagonist, was the first one to really weigh in on this subject because he was studying Australian and New Zealand megafauna, because these things are big. These things that disappeared are big. In La Brea, there are no small things from La Brea that disappeared. The birds are all still here that are from La Brea. The amphibians, all still here. The insects, all still here. I think there's one toad that is extinct. Meanwhile, all the big stuff is gone. So something about these megafauna that were disproportionately affected. And Owen recognized that based on studies of the moa of New Zealand. Moa, there ain't no moa. You know that one? Well, they're all gone too, as were a bunch of big vertebrates. And Owen speculated that humans, with dingoes maybe, with commensalistic dogs, contributed to their demise. He proposed that in 1877, and more or less in passing. Pretty quickly, someone said, no, humans can't have that kind of massive effect, and it must have been a climatic change that led to the demise of these big creatures. And ever since the 1880s, it's been humans or climate in this pendulum swinging debate within science. And it's a really interesting debate. It takes on different tones in America as it does from Europe and Australia. And I'm not going to spend much time on it, but you do well to be aware of it. Also, when you're just walking in California, thinking about how different the landscape is today from what it was that humans saw when they arrived. With regard to climate has undergone cycles of change, as evidenced by the carbon dioxide record and independent proxies for the temperature record over these hundreds of thousands of years. These are the ice ages and warming interglacials, and the ice ages and the interglacials, cycling back and forth. All these big creatures survived all these vicissitudes of change. It's up in here during a time when generally there was a warming trend evident, is when all this extinction occurred, in this window in here. Nothing really unusual about climate, as evidenced by this, relative to the deeper past. What is unusual about the timing is the correlation with people arriving. There seems to be a correlation between big things going extinct and humans arriving in different parts of the world. It's very clear on big islands like New Zealand or Madagascar. The debate centers on whether in a big place like North America or South America, whether that correlation represents causation. Correlation of human arrival, some causal effect on the creatures there. How can humans affect whole ecosystems in this way? The simple story is, well, spear in the gut, right? Spear in the gut, cook animal on fire, right? That's the overkill hypothesis. People just killed everything. What are other, thinking ecologically maybe, what are other effects that humans have on communities in addition to that? They change the landscape, they burn a lot of things. Fire is a hugely important piece in this puzzle. Humans are expert with fire, and the humans that arrived had fire kits and domesticated fire long, long before. And humans love to burn. Anywhere you go in the world, and you go at night, go up on a high elevation area, and you look around, you see fire, 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 fire. People burn stuff all over the world all the time. And they burn things at odd times of year relative to natural fire regimes. So that might be important. The role of fire in altering habitats, changing communities. What else? Agriculture. Yeah, agriculture is at play in the latest phases of this, but agriculture really didn't get a start until about 12,000, 10,000 years ago. So some of these extinctions predate that, but yes, there's clearing for agriculture of land, modification of habitats for growing things in the later phases. Yeah. Taking up resources, competition for resources. Maybe the humans are eating some of the same things the other creatures are eating. So an ecological competition for resources. Sure. What else? How about the creatures we bring with us? The parasites, the rats, things like that. Those are relevant. The diseases that people carry along with them. There's a multiplicity of things that humans bring in their wake that may be relevant to large-scale community changes. But again, it's a debate. Some people just really argue strenuously and convincingly against that based on tangible evidence. So explore it if you want. This diagram just shows the degree to which it's the large creatures that disappeared. A histogram of body sizes of creatures in North America that existed in the Pleistocene. The ones dark shaded are the ones that went extinct. They're almost all the big end of the spectrum. The big end of the spectrum. That's what this shows. It's more clear the fact that humans are causing massive change today and widespread extinction today. Those end Pleistocene extinctions affected the big vertebrates, the megafauna. It wasn't a mass extinction by any account, because a mass extinction by definition affects a broad array of organisms, not just one small category like the large vertebrates. You'll see in ecology, I'm sure, that the large vertebrates are extremely important in ecosystems, in shaping whole ecosystems through cascading effects of those vertebrates. But it's not a mass extinction. Today, there's an argument as to whether we're in a true mass extinction based on the work humans have been doing on the planet since the Industrial Revolution, say. Because you're losing fungi, you're losing amphibians, you're losing bats, you're losing marine flora and fauna, you're losing probably bacteria, while evolution is still continuing also. But there's a debate as to whether we're in a sixth mass extinction event now. We highlighted the first 5 that are obvious. Well, is this the sixth extinction that we're in the midst of? If you're interested, click here for popular sorts of articles on the subject or a technical article here. Some want to rename a new geological time for our time now as the Anthropocene. And that's gaining a lot of traction in the humanities and the sciences, and if you haven't bumped into it yet, I'm sure you will. It's become very popular, the notion, and geologically stronger and stronger, the arguments for a new geological time, the Anthropocene, anthro meaning human, this human-dominated time. To be a true geological time, you need a marker. You need something of a global marker in the geological record. Those plastiglomerates would qualify, but they're not showing up until the last couple of decades, these melted plastics conjoined with a bunch of other natural material. If you're going to date the beginning of the Anthropocene, an article just published in Nature, the world's leading scientific journal, argues pretty convincingly for 2 possibilities of dating the beginning of the Anthropocene, if you want a concrete date. One argument is for a date of 1610 in the Common Era, right? So just 400 years ago. And they're basing that on tree ring analysis that shows a tremendous decrease in carbon dioxide, a decreasing trend in carbon dioxide rapidly occurring here. And the relation is with colonialism, essentially, with European cultures expanding across the world and, in a word, decimating local human populations in the Americas and also in Africa, involving the slave trade and just a general collapse in numbers of humans around the world, leading to a reforestation of agricultural lands. So an actual increase, a great increase in trees and photosynthesis around the world as a result of human depressed numbers of humans. These details aren't important. This isn't even on your slide, this fact. But that's the argument for 1610 as a date. That would involve the decrease of carbon dioxide, because with so much photosynthesis occurring, so much carbon dioxide is sequestered, and you see a global collapse like that. So they say maybe 1610 is a good date for the start of that. Another argument is for nuclear weapon testing in the 40s and 50s, around World War II, that released carbon-14 into the atmosphere. Carbon-14, that radioactive isotope that's used for dating, well, it's been increasing. It increased in numbers tremendously around World War II with nuclear bomb testing. And that increase is seen in tree ring analysis, because trees take that carbon dioxide in, and you can see around 1964 in particular when trees started to incorporate it in bulk. So there's a geological signature based on botany, based on trees, for a massive change related to human activities. What's clear is humans alter the face of the planet in dramatic ways, shocking ways in terms of the scale of this effect. But we've seen other examples in the fossil record, right? What cyanobacteria did in the early history of the Earth. Other examples. I'll return to that cyanobacteria example in our review, because a couple people asked about that. So yeah, that's it for the materials for the section. So let's review some things. In the emails I received, there were several overlapping questions. If I don't cover something you emailed me about, I will email you back with a short answer related to the question you asked. I kept a list of those. And if I don't respond to you, I think I've got them all clear. So I should get to every one of them. But if I don't respond in the next day or so, just email me back. I'm not able to respond in detail to long lists of questions on email. It just takes too long. If you have long lists, feel free to send them, but I might say, can we do it on the phone, 2 minutes on the phone saves me 25 minutes on email. But please do first try to cover it with your classmates and in office hours. Office hours here, GSI office hours, they'll be held all right up to the exam. Here's a practice question that I got a couple questions about. I like this question because a couple things it illustrates. So which of the following is not a factor inference that Darwin relied upon in his work on evolutionary theory? A couple things about this. A, there's heritable variation among individuals. Certainly. Fundamental to Darwin and Wallace's view of nature, right? And fundamental part of the natural selection argument. There's a struggle for limited resources. Definitely. I didn't highlight it so much, but it's a big part of their argument. It goes back to Linnaeus, that there's a struggle for existence based on a struggle for limited resources. Based on the Malthusian principle of overpopulating areas with finite numbers of resources. So right off the bat, you've got two that are absolutely correct. Definitely. Individuals whose inherited characteristics best fit them to the environment will, on average, leave more offspring. Sure. It's fine. That's part of Darwin's model. Now you've got 3 out of 3. Offspring inherit characteristics acquired by their parents during the parent's lifetime. Wait, isn't that supposed to be Lamarck? And then you scratch your head. That's not right. And then you get to the last one. You don't want to miss the last one. All of the above. Oh, boy. Now I've got 3 and 1 not so much. Maybe I'm wrong about this one because I'm sure about those. Good. Go for E in a case like that, right? You're sure about these 3. You're sure. There's only 1 you're not sure about? You better be really super certain that it's wrong. And you might feel that way based on maybe high school biology or something. But I tried to emphasize the extent to which Darwin accepted inheritance of acquired characteristics. He did. He accepted it and even made it a bigger and bigger part of his evolutionary theory later in life. That whole idea of pangenesis, that inheritance mechanism he invented, was a Lamarckian, quote-unquote, Lamarckian model. So, E is a great answer there. All of the above. All right. There was from lab quizzes, I think, in particular, and then a couple of my practice questions from the new set. relate to a point about Hardy-Weinberg that I just want to make quickly. Whenever I do these diagrams on the board, I seem to put people to sleep. So I'm going to try to stay brief, and please stay awake so we can continue after this. But the question relating to genotype frequencies and allele frequencies in populations that undergo non-random mating, right? Does non-random mating change genotype frequencies and allele frequencies? I know that there were at least a couple sections that had that as an exam question. I'm going to try to turn the lights on up front on the board. And so I think I can illustrate this pretty easily. If you imagine 6 individuals, 6 parents, in a biallelic context, we're always dealing with a biallelic context for these models. 6 parents with 3 genotypes. Two of them are homozygous for the big A allele, 2 are heterozygous, and 2 are homozygous for the small a allele. So what are your allelic frequencies in this population? What's P, if P represents the frequency of A, right? Frequency of A. What is P? Who's got it? Go ahead. 0.5. Excellent. Because you have 12 alleles in total, 6 of which are big A. P equals 0.05. Q, well, you can figure Q out either by 1 minus 0.5 or doing the same thing. And what you'll find is it's also 0.5. So your allelic frequencies are 0.5, 0.5. And if you predict, based on Hardy-Weinberg, the genotype frequencies of the next population, P squared plus 2PQ plus Q squared, what are you going to get? P squared, 0.25, right? 2PQ, 0.25, 0.5, plus 0.25. And it better equal 1. It does. So your prediction in the next generation would be for your homozygous individuals of big A to be 25% of that population, your heterozygotes, and your homozygous for little a. Now, what happens if these guys are non-randomly mating? If they're mating assortatively, which is a positive assortative mating of like with like. Like phenotype with like phenotype. Organisms relate to each other based on their phenotypes. But we take that to represent a non-random mating of genotypes together, because those phenotypes reflect the genotypes. So if these guys are mating non-randomly, then your homozygous individuals for A are going to mate just with themselves. And your heterozygotes are going to mate likewise. So it's going to be this situation, right? Because they're going to mate with the same types of individuals. And if you do that, what are the offspring in the next generation of these organisms going to look like? So if we have the offspring genotypes, okay, how many are going to be AA? How many are going to be heterozygous? How many are going to be little a, little a? For these, all of them are going to be big A, big A. Your 4 offspring, say, of these individuals. Do a Punnett square with AA, and work it out. right? If you cross these two individuals, well, only one out of the four is going to be homozygous for big A. Two are going to be heterozygous. And one is going to be homozygous for little a. So a distribution like that. Across these guys, they're all going to be little a, little a. There are no other alleles in the mix here. So what does that mean for this group in the end? 5 of them, 2 of them, 5 of them out of the 12. 5 individuals, homozygous big A, 5 homozygous little a, 2 heterozygous. What are those percentages? I wrote them down because I can't do them in my head. The percentages are 0.42, 0.17, and 0.42. They're different than our predicted values for the genotype frequencies. There are more homozygous individuals than predicted for big A. There are more homozygous individuals than predicted for the little a. And there are fewer heterozygotes than predicted. So this non-random mating process led to genotypic changes in genotype frequencies. But count up the alleles in these populations, and you'll find that there's still half of those alleles are big A and half of those alleles are little a. So genotype frequencies changed, but allelic frequencies didn't change. Genotype frequencies specifically changed with an increased number of homozygotes than more than predicted. So this non-random positive assortative mating creates greater homozygosity. And that's one of the problems with inbreeding. With individuals who share so many alleles, there's an increased homozygosity, which is particularly troublesome if there are deleterious alleles that come into combination and you get a homozygous recessive condition that can lead to disease. But I need to review more stuff. So if you have questions, let's hit it in office hours, and so we can get to this one, because lots of questions on this. And it is one of my favorite diagrams with regard to speciation. So let's make sure we understand it. A group of populations here. These represent populations, 5 individuals in each population, right? And they're all interacting, even though they're populations, they're separate, and you have one meta-population of smaller populations. They're all interacting closely, and gene flow exists among the populations. So they're circulating alleles more or less freely. Some kind of barrier develops. Imagine a river, a creek, and then a larger stream, and then a river, beginning to isolate these two populations. As a result of isolation, different factors come into play and start to lead these two groups to diverge, one becoming more red than the other group. Gene flow still exists, but less of it. Or if you want to imagine this as a hard barrier, no gene flow at all for this time period, and then a bit of an opening up with a small amount of gene flow now occurring. So what you have at this position in history are two populations with a hybrid zone between them, where these hybrids are taken to be fertile and viable over the long term. Someone asked about that as a mechanism of exclusion. You can have offspring that are fertile, but that break down over the generations and become less and less viable. Let's assume these hybrids are both fertile and long-term viable, such that you have these 3 different morphs existing simultaneously. Others ask, well, is this 1 species or 3 species? Depends on your species concept. Most biologists would take this to be 1 species with 3 different morphs, subspecies, say. Subspecies A, B, and C. Now what happens in time, into the future? What are the possibilities of what may occur here? Well, these differences may continue to get reinforced if these trajectories continue to separate them. In this metaphorical sense, you could get a reinforcement of the differences and a breaking down of this hybrid zone because they're just not crossing anymore. And then you can imagine speciation happening. And they become two good, distinct species. You could also imagine that they completely come back together and fuse back together into a single, presumably original form. Maybe with some new differences based on the unique history in here, but resembling that original form. Fusion, back to a single species. Or you just carry on like this, stably, with a hybrid zone, two other morphs, and there continue to exist three subspecies like that, which seems to happen over long periods of time for some organisms. And it's a possible eventuality, more or less permanent for the organisms until something else affects them. Any questions on that? Yes? How is reinforcement different from stability? Reinforcement is implying that the differences that exist continue to accrue and making gene flow lower and lower between the two, such that this hybrid population eventually ceases to exist. So the thinning of that green line is to symbolize that that's going to be reduced to zero. So the hybrid zone is going to go away. Questions specifically about this?
So where would Darwin's finches fall on this scale? I mean, their height is
Darwin's finches and the whole island hopping model is really a bit different from this, because you're thinking of strict allopatry there, without gene flow leading back and forth across islands, at least in the way I presented the island hopping model. Darwin's finches in actuality have more complexity than that, but the only way that I addressed Darwin's finches was in a strict allopatric island hopping model. The geological time scale, which I understand is one of the scariest parts of this section, just in terms of information content. So I want to limit it as much as possible to these dates and these events. And the point really is to get some sort of scaffolding in your mind for the geological history. You need just a few set points in order to understand the time scale. And those set points are these. Starting with so these arrows and times are repeated in this context, right? Starting with the origin of the Earth, which you learned in the last section too. It's not quarrel, 4-5, 4-6, whatever. Changes as science moves along, but somewhere around there. 3-5, what's this? These are the first fossils, right? Signaling the first big communities of prokaryotes in the form of stromatolite fossils. 3-5. The oxygen revolution, which I'll talk about in a second. 2-7, maybe 3.0, based on new results, somewhere right there. I'm not going to ask you what was the date of X. I'm not going to ask that. That's just a factoid. But it may come into play with relative timing. Or you might need a general sense. Well, this is 3.5 billion years ago, not 3.5 gazillion years ago, or 3.5, thousand years ago. So just that, a sense of that scale is important. The Cambrian explosion, this crazy radiation of animal phyla, all of which continue to be represented, or everything today is represented during this radiation. There are extinct phyla. And then human evolution, included up here at 2.8. The stuff I was talking about today at the end of the Pleistocene, it's way up at 60,000 to 10,000, that range. And then talking about, the last couple hundred years. Way up in a vanishingly thin slice of that portion of the 24-hour clock, the history of life. Any questions on those points? I'll talk about the oxygen revolution if you don't have any questions on it. Yes. Oxygen revolution. Now, this is in relation to, this is after the evidence for the first origin of life, the origin of stromatolites. That's the origin of life, the origin of the first organisms. Well, they do their thing. They're cranking along. And particularly in the form of photosynthetic prokaryotes like cyanobacteria, in the splitting of water molecules in the photosynthesis process, oxygen is released, right? Carbon dioxide is taken in, water is split, and oxygen, O2, is released during photosynthesis. If that's happening on a vast planetary scale, there's a bunch of oxygen being released into a world in which organisms don't use oxygen in their metabolism. Indeed, in a world where organisms find oxygen poisonous. So all this oxygen is being released, and at first is released into the oceans. And oxygen in the oceans reacts with iron, which is present in the oceans, and forms iron oxide, rust. And that settles into the sediments and is visible in the sedimentary layers from this time period. These bands of rust signal free oxygen reacting with free iron as a result of this explosion in photosynthesis. So the oxygen revolution is indicated by these bands, just like the human revolution is indicated by bands of plastic or bands of carbon-14. All right. The hominin fossil record. What do you need to know for the hominin fossil record? You need to know the details of every single thing I talked about. No, wait. As my daughter pointed out with a joke this morning, it's April Fool's Day, isn't it? Yeah, right. She told me we were having pancakes for breakfast when I was getting ready, and I got all excited. April Fools. All right. Yeah, by the way, spring break for my kids is next week, so if you're at office hours, excuse me, but they'll be distractingly in the corner somewhere. For the fossil record, let's say that the exam next week will not cover that lecture in any detail. Maybe a question, all right? Maybe one relatively simple question. I don't want you to abandon that stuff. Don't put it all off till the end. I'll ask you one basic question related to great apes. Nothing with regard to the later hominin fossil record, all right? So you can basically put that off till the end. But I hate to say that because you're going to just create work for yourself on the end of the semester when it's going to be so busy for you. All right, but just one relatively straightforward question on primates and great apes. Nothing on these guys. But in the end, you'll need to know these genera. Sahelanthropus, Ardipithecus, Paranthropus, and Homo. Australopithecus being this major genus in between. So you need to know something about what I said on those. Habitat preferences, the earliest form, the foramen magnum, Ardipithecus with its long fingers and divergent big toe, Paranthropus with its massive chewing apparatus, Homo and all of our craziness. Something about those phenomena. Okay. Thank you, everybody, and enjoy ecology.